So when we're talking about hair cells, what happens is when a hair cell is at rest, you have these cilia that are extended upwards. The tips of these cilia are connected together by linkage proteins. And what will happen is these tip linkages that are held together by these proteins, these consist of ion channels that are pretty much closed, but they might be open just a tiny little bit. So some positively charged ions like calcium, potassium might sneak in. This will cause a release of neurotransmitter at a very low rate. Glutamate is the neurotransmitter that gets released, and that causes just a few action potentials to be triggered in the connected afferent neuron. Now, when the hair cells, or the stereocilia of the hair cells are deflected this way, this opens up those ion channels in the tips of the cilia. This causes a large flux of positively charged ions into the cell. This influx of calcium depolarizes the hair cell, causing a large release of glutamate, which triggers a high frequency of action potentials in the connected afferent neuron. Now, if it's bent the other way, this closes off all of those ion channels, causing which stops the release of the neurotransmitter glutamate, which causes no action potentials in the connected afferent neuron. It's important to note that the depolarization that occurs in the hair cell, that's a graded potential. So how much the hair cell depolarizes depends on how wide these ion channels are opened, whereas the connected afferent neurons, they exhibit action potentials, the all or none kind of a signal of the nervous system. So what happens is, if we have like a low sound that we can hear, a low pitched sound that we can hear, this low pitched sound will travel down the vestibular duct, and it'll encounter a part of the cochlea where it just happens to be tuned to kind of the conditions of this part of the cochlea. It causes this tympanic, this membrane separating the vestibular and cochlear ducts to depress. When this part of the membrane is depressed, it stimulates these hair cells, okay, and that triggers action potentials in the connected afferent neurons to travel through the oculomotor nerve through to the ear, to the brain. Actually, it goes to the medulla and then the brainstem, and then it passes through the thalamus and finally to the primary acoustic cortex of the cerebral cortex. And that allows us to hear. But what's important is that the brain, like at this part of the cochlear duct is stimulated, these hair cells, the brain knows that those afferent neurons were connected to hair cells in this part of the cochlea, and that's how the brain knows it was a low-pitched sound. On the other hand, if it was a really high-pitched sound that comes in here, this high-pitched sound is going to be tuned to a different part of the cochlea, be tuned to depress this part of the cochlear duct. These hair cells that are located here will be stimulated, causing action potentials to be generated into those afferent neurons, and the brain knows that that's coming in as a high-pitched sound. And that's basically how the brain is able to, or that's how the ear transduces vibrations into action potentials. Now, on the other hand, what we have on when we're talking about balance and we return to these structures, the saccule and the utricle, the saccule and the utricle, they contain a different type of structure that also consists of hair cells called maculae. So I'll draw a little structure right here. And this blue structure that I just drew, housed within inside the saccule and the vestibule and the utricle of the vestibule, these guys are called maculae. And what maculae are is they're slightly different kind of organization of hair cells. You'll have a series of hair cells that are located underneath kind of a ceiling of rocky-like structures. This rocky-like structure is called the otolith. Okay, and what happens is we experience linear acceleration. So if a car stops, or if we tilt our heads, this otolith will move back and forth. This will cause the fluid underneath the otolith to displace the cilia of these hair cells. When that happens, it triggers action potentials connected to the connected afferent neurons. Now, the otolith located in the maculae of the utricle, which is right here, is oriented laterally, and so this is going to be stimulated if your car slams on brakes or takes off at a high rate of acceleration. It responds to acceleration in this linear dimension. Whereas so this guy is going to be in the utricle, whereas the macula in the saccule is going to be oriented vertically. So you have the hair cells here, and you have the otolith running beside it like this, and this is going to respond to up and down accelerations as if you were in an elevator. All right, and it can also detect the position of your head. So the position of your head in addition to linear accelerations. Now we have similar like structures in the base of the semicircular canals. These guys are called crista ampullaris. So these are bundles of hair cells in the bases of the semicircular canals, and they are called, draw it down here, crista ampullaris. And basically these arcs or these arches contain fluid, and as we experience rotational acceleration, fluid will flow through these arches, and it will stimulate the hair cells in these organs called the crista ampullaris, and those allow us to feel rotational acceleration, like if you're on a merry-go-round. This is our own natural internal gyroscope. We have arches, semicircular canals that can sense acceleration in 3 axes of motion, allowing us to feel acceleration from side to side, back and forth, and then spinning laterally.